Hello everyone, my name is Zhao Xiaoxian. Today, I will introduce our study published in iMeta, entitled Hyperoside Alleviates Endometrial Stromal Cell Senescence in Unexplained Recurrent Spontaneous Abortion via DHX9-mediated R-loop Resolution. Decidualization deficiency is a key pathological mechanism underlying unexplained recurrent spontaneous abortion, yet its definitive molecular mechanisms and effective therapeutic strategies remain elusive. Using multidimensional research approaches, this study reveals that hyperoside may target DHX9 to regulate R-loop stasis, alleviate senescence of endometrial stromal cells, and thereby ameliorate decidualization deficiency. First, our study found that hyperoside could reverse the pathological damage of decidua in RSA mice in a dose-dependent manner, upregulate the expression of decidualization markers PRL and IGFBP1, and reduce the embryo absorption rate in a dose-dependent manner. Next, we found that stromal senescence markers were increased in decidual tissues from RSA patients and mice. Hyperoside reduced senescence-related signals, including gamma-H2AX, P53, and RB, and decreased SA-beta-gal-positive THESCs, indicating a clear anti-senescence effect. To identify the upstream trigger of senescence, we performed single-cell RNA sequencing. Differential genes were enriched in DNA damage response and nucleic acid structure-related pathways. Further validation showed that R-loops accumulated in RSA decidua, while hyperoside reduced this abnormal accumulation. Mechanistically, R-loop accumulation increased cytosolic DNA and activated the C-gasting pathway, thereby promoting stromal cell senescence. R-loop clearance by RNase H1 mimicked the protective effect of hyperoside, whereas restoring R-loop levels weakened this protection. We then identified the direct target of hyperoside using LipMS, molecular docking, molecular dynamics simulation, SPR, and CETSA. These analyses showed that DHX9 is the key target for R-loop regulation, and THR419 is critical for high-affinity binding with hyperoside. To validate DHX9 function, we performed knockdown and rescue experiments. When DHX9 was depleted, hyperoside could no longer clear R-loops or suppress C-gasting activation and senescence. Only wild-type DHX9 restored the protective effects of hyperoside. In vivo, we further confirmed that the therapeutic effect of hyperoside depends on the critical DHX9 residue. Wild-type DHX9 rescue restored decidual structure, reduced senescence, and lowered embryo resorption, whereas the mutant failed to produce the same benefits. In summary, this study identifies hyperoside as a natural DHX9 modulator. Hyperoside targets DHX9 to resolve pathological R-loops and suppress C-gasting-mediated stromal senescence. These findings reveal R-loop-induced genomic stress as a key mechanism in RSA and suggest a distinct therapeutic strategy.